So my name's Cara Froggatt, and I'm going to be chairing the next section of the event. So during this session, we're going to hear a series of short talks from some former plant health undergraduate studentship students and supervisors who are going to be sharing the projects that they've been working on as part of their studentships. These studentships are an opportunity through the summer for students in the middle years of bioscience degrees. They get to find out what it's like to contribute to a research project and gain some valuable experience. Every year there are 12 studentships, and the programme is a collaboration between various different funders, including the Department of Environment and Farming and Research, Defra, the British Society for Plant Pathology, the Association of Applied Biologists, the SCI Horticulture Group, and the Colgreve Seabrook Foundation. So the aim of this session is to highlight the studentships, the work that came out of it, and the ongoing kind of projects from the supervisors and students. There's not going to be any question and answers during the sessions. However, most of the speakers have contact details available on the slides, which will be shared afterwards, and you're encouraged to reach out after today's session to ask any questions that you have. So without further ado, I'm going to introduce our first speaker, Vasili Tsourekas, who's going to tell you about his work.
Okay, thank you for having me. Hi, my name is Vasili, and I'll be presenting the work that I did with the Royal Society of Biology studentship at Harper Adams University, where I compared morphometric and molecular identification with agriotes, also known as wireworm. So to start with some background information, wireworm are click beetle larvae, which are polyphagous soil pests. So they eat a variety of different crops. And they're found in arable soil in North America, Europe, and especially in the UK. But most importantly, the most economically important species are found in the genus Agriotes, since they're the main ones that do the direct and indirect damage to crops, making them unmarketable and susceptible to other diseases and pests. Now, previously, wireworm were easily controlled, and they weren't as such as a nuisance. However, currently there's been a growth in their population, mainly due to the banning of organochlorines, which was their main control method. And also further research has highlighted that cropping and cultivation methods and even the changing climate has contributed to their growing population. So because of this, we need to come up with a new control method. However, before we can do that, we need to come up with a proper identification system, since currently there's a lack of research, especially on the molecular side, in identifying these species. And it's really important since these species have species-specific traits which influence their feeding behavior and phenology. So we need to properly identify which species we're dealing with to employ the proper control method that's specific or most effective towards that specific organism we're dealing with. So with that in mind, this led to the aims of the research, which was to identify agriote specimens that were sampled from East Wales through molecular and morphometric identification techniques, and then compare between these two methods. But most importantly, set up the molecular protocol for work with wireworm at Harper Adams University to assist with future research. So to start off, as I mentioned before, the two main techniques that I did was morphometric identification, which was distinguishing between species through their anatomical parts, and then DNA barcoding using segments of DNA, in this case, the COI gene as a universal barcode to molecularly identify these species. I specifically only looked at 5 of the 9 economically important species. So it's Obscurus, Lineatus, Proximus, Spitator, and Brevis, since these are native to the UK. So to start off with morphometric identification, I did this through discriminating characters. So they can be divided into primary characteristics and secondary characteristics. Primary, just an anatomical trait that's only seen in 1 or 2 species alone, giving an 80% chance that you got the right identification, while secondary characteristics just gives that extra evidence for proper conclusive identification of the specimen. The specific anatomical parts that I looked at were the mandibles, the clippus, and the ninth abdominal segment, as you can see in the two figures on to the right. And this was done using a dyno capture system. And here's the setup that I had. Um, but what made this system really good was that I could not only take pictures, but also actively measure specifically the different parts. So here's an example of me measuring the mandibles and the angles to make a more conclusive identification in the end. So for molecular identification, after I was done with the morphometric, I extracted DNA of the 22 specimens. And then they went through 3 different batches of PCR with 3 different sets of primers for the following species. And then 3 different batches of gel electrophoresis in order to see if there was a presence or absence of DNA. But if there was presence, I specifically looked at the base pairs to be able to accurately pick which species was present for that specific specimen. So first of all, the results for the morphometric analysis was most were Agriotes obscurus, which was expected since in that location, the most wireworm population is of that species. However, what was more interesting was that a couple of the samples were unidentifiable. This is mainly due to the anatomical parts, such as damaged mandibles. As you can see in the image here, it's really hard to actually properly measure that, so it wouldn't make a good conclusive identification in the end. So in the end, I just left it as unknown. For the molecular identification, more species were found to be obscurus, which was again expected, with 4 samples being lineatus. However, 0 samples being proximus, brevis, and sputator in the end. Now, when comparing both methods, it's seen in biological research that molecular identification is a more reliable and faster method. This helps even just further with genetic research and identifying cryptic species, but it also can be added on a wider scale with multiplex PCR. But again, there is still a higher chance of contamination and just an overall expensive method. But when comparing to morphometric, it's just really difficult and it becomes to a point where it can be undistinguishable between species, especially ones that are really closely related, such as Agriotes lineatus and Agriotes proximus, which their morphology alone cannot be help identify between the two. And then obviously there are higher chances of finding identifiable samples. So the contributions of this specific research has helped further improve other research at Harper. So first, it's assisted with 2 experimental papers, one that's going to be under review for publishing, but most importantly, started a protocol for the entomology lab for molecular work with wireworm, and it's actually formed the basis for a project in fumigation and entomopathogenic fungi, but it also contributed to a PhD application, which will be looking or investigating controlling wireworm in arable crops. My overall studentship experience was actually amazing. I got to contribute in a variety of different activities that a normal PhD student would be a part of, such as going to a potato demonstration day, where I got to actually interact with farmers and hear their issues not only with wireworm, but with a variety of other pests and diseases. I also got to present at a plant health symposium and also got to present my research through a poster as well. But most importantly, I got great advice and insight on my future through the PhD students that I met, but also my supervisor, who helped me kind of guide of what I want to do in the future and if a PhD is the right fit for me. So in the end, I did decide to apply and I did get accepted to one where I'll be starting in September and October with the SW Biodoctoral Training Program, where I'll be looking at the impacts of climate change on Fusarium head blight disease and mycotoxin production in wheat. And I'll be doing that with the University of Exeter and University of Bath, also collaborating with Syngenta. So yeah, thank you.
Thank you very much, Vasili. That was really interesting to hear about what you're researching, and it's great to see what you're doing next as well. So our next speaker is Abby Buckle, who is a student from University of Sheffield. So Abby, if you want to start your talk and share your slides.
Brilliant. So I'll just share my slides. Are you able to see my slides?
Yeah.
That's great. Brilliant. So I took part in the RSB studentship program for 10 weeks last summer. I was based at Rothamsted Research, and I was working on a project looking at oilseed rape resistance to cabbage stem flea beetle larvae. And my studentship was supervised by Patricia Ortega Ramos and Sam Cook, and with help from Alex Dye. So just some quick background on cabbage stem flea beetle and OSR. So oilseed rape is a hugely important crop, both for vegetable oil and biofuel, but it's currently under threat from cabbage stem flea beetles. So both the adult and the larval stages of the beetles are damaging to the plants, and they were previously controlled by neonicotinoid seed treatments. But since these have been banned, there's now only one registered insecticidal control group, which are pyrethroids. And unfortunately, the beetles are showing a growing resistance to these due to the lack of alternatives and the high spraying frequencies. So it's really crucial that alternative, environmentally benign pest management strategies for cabbage stem flea beetles are developed. So my project was part of a wider research project that's looking at developing integrated pest management strategies for cabbage stem flea beetles. So I focus specifically on the larval stages, and the larvae feed within the petioles and the stems of the plants, and they impact things like crop vigor, reducing overwintering success, and decreasing the plant's frost tolerance. So my project aimed to identify cultivars of OSR that are more resistant or tolerant to cabbage stem flea beetle larval damage. And to do this, we tracked larval infestation and development across different OSR cultivars in field conditions and looked at any associated impact this had on plant growth and yield. So as I mentioned, we were looking at commercial OSR lines. So we had 20 lines in field conditions, and there were 5 plots of each line, and you can see those here. And once a month, 3 plants were harvested from each plot throughout the 2022-2023 growing season. When they were harvested, the plant characteristics were recorded, which included things like height, weight, and growth stage. And the larval infestation was measured using drying evacuation methods. So this is when the plants are left to desiccate, which again, you can see here. As they dry out, the larvae evacuate the stems and they fall into the trays below, where I was then able to count the number of larvae and identify the different instars. So there are 3 larval instars for cabbage stem flea beetles, and I was able to see which development stage they were at. And then at the end of the season, all the oilseed plots were harvested and the yield was recorded for the different lines. So then from this, I was able to compare the number of cabbage stem flea beetle larvae in each line at key points in the growing season. So I focused on from data from November, January and February, as these are the points when the larval damage is expected, the plants are expected to be most vulnerable to that larval damage. And here you can see, so I looked at the number of total larvae and also the number of third instar larvae. And I wanted to drill down specifically to look at third instar larvae, as they are the largest and expected to be like the most voracious feeders and the most damaging. So you can see that there were lines that had significant differences in larval numbers. So you've got 1 line in November that had significantly more total larvae, 3 lines in February with significantly fewer total larvae. And then with the third instars, again, we've got 1 line in November with significantly more, and then 5 lines in February with significantly fewer. In both cases, I didn't find any significant difference in the data from January. It is worth noting here that the lines that have the significant differences in total larvae aren't necessarily the same that have the significant differences in the third instar larvae at the same time. So for example, total larvae, line 19 had significantly more total larvae in November, but actually looking at third instar larvae, it was line 5 that had significantly more significantly more third instar larvae in November. And that's something that I will come back to. But the main aspect that I was focusing on was whether these significant differences had any impact on the grain yield at the end of the season. So here we've got, again, those larval numbers and then the grain yield here on the right. And there were 2 lines that had a significant difference in the grain yield. So lines 8 and line 12 both had higher grain yield than the other lines at the end of the season. And looking at how this relates to the number of larvae, I've highlighted those lines here, and you can see that neither of those lines had a significant difference in the number of either total or third instar larvae. And actually, those lines that did show the significant difference in the number of larvae didn't have any significant difference in the grain yield. So looking at kind of what can be drawn from that, as I mentioned, those lines that had the significant difference in the number of total or third instar larvae at the stages I was looking at didn't show a significant difference in the grain yield. However, those lines that showed a significant increase in the yield, despite not having that difference in larval numbers, may indicate that they're more tolerant to larval damage and larval feeding, so they may be able to maintain that higher yield even with a higher number of cabbage stem flea beetle larvae. And this is something that may be able to be included in integrated pest management strategies, giving growers a choice of cultivar based on that tolerance. And as I mentioned as well, plant characteristics were recorded when the plants were harvested. So it's also an avenue for future research, looking at what characteristics could potentially increase that larval tolerance. And then finally, going back to those lines that had a significant difference in the total larvae, but not the third instar larvae or vice versa. This may indicate that there's a difference in larval development between the lines. And again, maybe an avenue for future research, looking at whether the cultivars are impacting at which stage the larvae are reaching those different instars, and again, how that impacts feeding damage and grain yield. So that's a summary of my project in general, but just a bit about the studentship. Being able to take part in the RSB Plant Health Undergraduate Studentship was a hugely valuable and hugely enjoyable experience. And I was able to get really great experience both in the lab and out in the field. I was able to go out to the farms and meet the farmers who were growing oilseed and see a whole wide aspect of the research project. And it was, just really great to be able to see that, and a really great introduction to integrated pest management and the research that's going on around that. And on top of that, I was also invited by the Association of Applied Biologists to attend their IPM and biocontrol conference, and if I wanted to bring a poster of my work. And this again was a really valuable experience, really useful. It was really great to see a whole range of research that's going on and meet loads of people working in the field and get a really good idea about what I wanted to do going forwards. So I just wanted to say a huge thank you to Patri, Sam and Alex and everyone at Westhamstead, as well as the RSB Plant Health Undergraduate Studentship Programme and to the Association of Applied Biologists. Thanks.
Thank you, Harriet. Thanks very much. Hopefully you enjoyed the conference that you had the opportunity to go to as well. Our next speaker is Dr. Tom Pope, who is one of the FLUG supervisors based at Harper Adams University.
Good. Well, thank you ever so much for the opportunity to speak to you today about our experience of hosting a summer studentship last year. And that studentship fitted into a wider programme of work at Harper Adams, developing sustainable approaches to the management of aphid BYDV vectors. And so I just wanted to take the opportunity today to just kind of place that into context with our wider activities and maybe to say just a little bit about Harper Adams, and for those who are less aware. So we're a specialist land-based university based in the West Midlands, not far from Birmingham in the county of Shropshire. Because we're land-based, we're fortunate enough that the campus is situated on a commercial farming enterprise based on mixed livestock and arable, consisting of just under 500 hectares. The entomology provision is centred around a bespoke entomology laboratory, the Jean Jackson Entomology Laboratory, and then associated glasshouse, polytunnel and field trial experimental areas. At the university, we offer a range of courses, both for undergraduate and postgraduate. At undergraduate, we offer an undergraduate degree in zoology with entomology, and then postgraduate courses in entomology and integrated pest, weed and disease management, amongst other options. The entomology group is broadly split into two, both focusing on ecological as well as applied entomological research with very much a crops focus. And the applied entomology research really seeks to develop solutions to promote best practices for crop protection globally. So that's very nice to say, but what does that mean? Well, our focus is really around developing effective biological control solutions. So that's where we're trying to exploit the natural enemies of key crop pests. We're focusing, particularly relevant to this studentship, on plant-insect interactions. So that may be how the pest damages the crop, but it may also be focusing on host plant resistance to the pest. And an increasing area of interest in modern agricultural systems is the replacement of those synthetic pesticides with biological solutions, particularly biopesticides, which are typically based on microbial as well as natural plant extracts, and using those in place of those synthetic products that we've previously relied on in agricultural systems. So that's the background. Specifically, the summer studentship focused on the management of barley yellow dwarf virus. And so, quick introduction to barley yellow dwarf virus, or BYDV. It's a very widespread, economically important plant virus affecting a range of cereal crops such as wheat and barley. It's primarily vectored by aphids, particularly in the UK by two species of aphid, hence the entomological focus here, the English grain aphid and the bird cherry oat aphid. So the aphids bring the virus into the crop, and then they spread the virus within the crop. And plants that become infected with the virus take on a very characteristic coloration, where the tips of the leaves become yellow and then sometimes bronze in color, reddish bronze coloration. And if you take a step back within the crop, you'll see areas where the plants have become stunted, and in some cases actually killed by the virus if the infection happened early enough and was severe enough. All of this means that the barley yellow dwarf virus is causing significant yield losses in UK agriculture and indeed agricultural systems throughout the world. In the UK, for example, we can see yield losses of up to 60%, and it's estimated that the industry loses around £136 million a year. So really significant economic impacts. Much of what this research effort has focused on really stemmed from some discussions with some colleagues who worked in this area many years ago. In fact, they began their careers back in the 1970s and 80s. And they began their careers back then because aphids were a huge problem in winter wheat crops. And that was a time when a particular variety called Maris huntsman was being widely grown throughout the UK. And both Professor Simon Lever and Keith Walters began researching aphid vectors of barley yellow dwarf virus in relation to Maris huntsman. But we were interested because when Maris huntsman stopped being so widely grown, the aphid problem subsided. It didn't go away, but it did improve to some extent. And so that led to the first student project, this one carried out by a Harper student, Dan Hawes, who wondered if Maris huntsman is actually preferred to modern wheat varieties. Obviously, it's 40, 50 years ago since Maris huntsman was being widely grown. How does Maris huntsman stack up against modern wheat varieties? We have a perception that modern varieties are bred for yield and quality at the expense of susceptibility to crop pests. But was that true? So Dan took winged or alate English grain aphids and simply released them from the center of an arena, and then presented them with different varieties of winter wheat, as well as ancient wheats from which those modern varieties have been bred, to see where the aphids prefer to land. And the result was really quite striking. Dan found that we have 20 times more winged aphids landing on Maris huntsman than on some, shown in the blue bar here on the right-hand side, than in some of the modern recommended list varieties shown over to the left in the salmon pink color. Also, quite interestingly, some of the ancient wheats also had relatively high numbers of aphids landing on them, giving some cause for pause, I suppose, on that general assumption that modern varieties are more susceptible than their ancient ancestors. So this led on to a PhD project that is being carried out by Maria Damascena. And what we were delighted to be able to do last year was to welcome Charles Bush through the summer studentship program to Harper Adams to work alongside Maria on this research activity. And really what we were trying to do as part of this project is to grow Maris huntsman around a wheat crop. So the idea here, the concept here, is that if Maris huntsman is such an attractive host plant for aphid vectors of barley yellow dwarf virus, can we intercept them before they make it their way into the main crop. So by protecting the main crop, and then we can manage the aphids in the surrounding area around the crop, close to the field margins. And if we can do that, we have an opportunity to combine our variability in host plant susceptibility with conservation biological control. If we're pulling most of the aphids towards the field margins, well, then we can enhance the numbers of natural enemies in those field margins to predate and parasitize those aphids that would otherwise be bringing the virus into the crop. And what Charles contributed to was that ongoing research program, and indeed that's led on to the final part of that program, alongside the trap cropping element. We're also interested, if the winged aphids bring the virus into the crop, well, then the wingless aphids, once in the crop, they then spread the virus, what we would call secondary spread of the virus, to the surrounding plants. But the secondary question here is, what happens if you increase the genetic diversity of your crop? Rather than growing a single crop variety, what we would refer to as a monoculture, what happens if we grow the different varieties in an intercropping or as a varietal mixture? What does that do to the spread of the aphids and therefore the spread of the virus? And the spoiler alert is rather that it does have an effect on the spread of the virus. So that's an exciting area that we're following up on at the moment. That's probably all I've got time for this morning, but hopefully it gives you an idea of how Charles was able to feed into this established research program and make a really valuable contribution to it. Thank you very much.
Thank you very much, Tom. That was really interesting. And our final talk of this session is Harriet Foster from the University of Bristol.
Yeah, just going to try and share my screen. Okay, hopefully that's all showing.
Yeah, that looks fine.
Okay, great. So yeah, I'm Harriet, and last summer I had the opportunity to work in Professor Diane Saunders' lab at the John Innes Centre in Norwich, looking at the future threat of wheat stem rust in the UK in a changing climate. So as a short introduction, I was inspired to apply for FUGS because I was really interested in plant pathology from a module I completed in my second year at university, but wanted to expand my research experience and academic connections before I truly considered a career in it. I found this a really helpful opportunity to both forge these connections, but also feel like I'm making a valuable difference in the field of plant pathology. So with that in mind, I'm going to be talking a little bit today about some of the outcomes and methodologies I developed throughout the project in the context of how this advanced diagnostic and modelling research in the Saunders lab. So first, a short introduction to wheat stem rust. Wheat rusts are often called the polio of agriculture, and wheat stem rust specifically is this biotrophic fungus which causes these raised orange-red pustules you can see here. It can be really aggressive and cause major yield losses. And with this in mind, in the 1950s in the UK, there was a massive removal program, which actually successfully led to it not rising in the UK. However, recently, around 10 years ago, we began to see this emergence of these more aggressive strains, which many of our UK wheat cultivars aren't resistant to. In fact, I think a paper came out last week, which actually suggested with a 2022 wheat stem rust isolate, only around, I think, 96% of wheat cultivars were susceptible. So there's definitely a need for rapid diagnostic and forecasting pipelines to ensure a targeted response. And this was what my project was focused on. So one of the key issues with ensuring that you're working with successful infection assays and actually creating the right output is just ensuring you're working on the right species. So I've put up a picture here of wheat leaf rust, and as you can see visually, the spore emergence shows very similar patterns. So visually, it's very difficult to distinguish. So there's a need to shift towards genomic resources which need to be developed. Secondly, you need to consider the favourable environment. So in the disease triangle, we know that we have a susceptible host and we know that we have a pathogen, but we need to think whether the environmental conditions could be conducive to an aggressive re-emergence in the UK. And this is particularly important in the context of climate change, where certain temperatures can induce life cycle stages of wheat stem rust emergence. So with this in mind, my two aims were firstly to trial and develop the wheat stem rust extraction and diagnostic pipeline, and secondly to use wheat cultivar inoculation assays in slightly more variable greenhouse temperature conditions to monitor lesion traits and biomass accumulation over the asexual portion on the primary host of the wheat stem rust life cycle. So firstly, for the first aim, I got to trial new novel genomics techniques, which I previously hadn't had an opportunity to do in university due to COVID. And this was primarily focused on primer trials at the PCR and Sanger sequencing level. However, this later was carried forward into phylogenetic tree creation and reinoculation with samples once I'd successfully discriminated them and ensured that I was working on the correct sample set. So I worked on ITS, TEF1 and PGTG primers. I first trialed this at the PCR level, looking for early band discrimination, both with different temperature and dilution assays, and then carried some forward for MySeq sequencing. Although I found that 86 ITS was really strong at PCR level, the forward sequence was quite poor for sequencing, so then I developed this further and eventually found that 48F was good for an alignment. And then this can be visualized here, where even an unaligned sequence, you can see I successfully identified that some of the samples we were using in infection assays actually weren't the correct species. They were PT, which is weak leaf rust. So this could then be removed for future assays. However, if you then add the alignment from the optimized pipeline I created, you can see the phylogenetic tree is slightly more advanced, therefore suggesting that I can produce a discriminatory and aligned sequence with this pipeline I've created. So for the second aspect of the project, I was working on a specific PGT or wheat stem rust isolate called UK01, and I was monitoring its aggressiveness over 3 cultivars, Siskin, Crusoe, and Vuca, which are all commonly used within the field, although it's varying a bit now depending on their resistance. And I was trying to see also whether there could be a correlation between visual monitoring and internal correlation, so we can create a corroboration between more genomic and more visual techniques. So firstly, I did a more subjective approach where I was just monitoring visually using this arbitrary scale. And then I carried this forward to plot disease progress curves through time following inoculation and plotted this with a fitted logistic growth model. As you can see, the 3 cultivars I trialed all showed very similar patterns of Uridinea spore emergence, which is the sporulation patterns through time, suggesting that with this specific strain, there's a very similar pattern of infection. And if you also take this into consideration that I did this in more variable greenhouse temperatures, which are definitely less controlled than what you might get in a growth chamber, you can see that the infection patterns are still quite aggressive and continual, which suggests that maybe there needs to be an introduction into these temperature assays to account for how there may be more environmental variation. So in conclusion, there was no significant difference between these trialed cultivars, but it should be considered that there was consistent Uridinea spore emergence. And this can be also viewed in light of climate change and potentially how even a broad range of climates is conducive to its emergence. However, you might be thinking that that was quite a subjective method. So I also got to trial the Plyman package, which uses entrainment through the algorithm. So it looks at different things of color. So I can produce a more quantitative plot, which I've got here, which also shows the visual differences through time. So as you can see, it produced a similar pattern, but was slightly more diverged between cultivars. And then I also wanted to see whether this could be mimicked in a more internal analysis, and I did this using qPCR. This hadn't been completed for fungal biomass in our lab before, so I got to generate a new protocol, which I did independently. And you can see that it shows very similar patterns, but it's a much more gradual increase. But if I just blow it up, you can begin to see how these patterns emerge. And then this was also very interestingly did show a corroboration with the Plyman plot, which I've put up here. So there is a correlation between how fungal biomass accumulates within the leaf with wheat stem rust and how it can be visually interpreted. So that suggests that it would be really useful to integrate a genomic and a visual approach. So these are my conclusions throughout. It was really interesting to reach back out to the lab and to sort of see how my research had sort of been continued on. So for example, with the optimized pipeline, all of the purified and bulk samples I created are now being applied in inoculation assays. And my optimized pipeline is now being applied for samples which are being sent in this year by farmers. So it's a much more rapid diagnostic method. I also thought it was interesting how now my supervisor is continuing my temperature research with more cabinet-based temperature assays, but it's accounting for the fluctuations in daily temperature. So this was a really interesting bit of early preliminary research. I thought I'd just end with a short reflection on careers and sort of how FUGS has helped guide me. So I finished my undergraduate degree last week, and then next in August, I'm beginning a Master's by Research on Zymoseptoria with Dr. Andy Bailey and Dr. Pierre Bouscaille, specifically looking at the physiological and molecular aspects of sporulation patterns in relation to airflow. And obviously this is another fungal wheat pathogen. So I found that the FUGS program really helped me hone in on exactly what I found was interesting. And for that reason, it was such a valuable experience, and even moving that forward into the future. I think is something which I want to carry forward. But I'd also like to credit other projects I've been part of throughout my degree. For example, my work with Better Cider and Thatchers, looking at infection patterns in orchards and how that can affect industrial output. And I've also been working on a literature review on the decline of wild bees, which has really helped me broaden my environmental context and help me sort of perceive how I can carry through my research into a broader scale, adapting it to a more widespread audience. So yeah, I would just really like to thank folks for allowing me this opportunity to sort of move forward with my research and to get to see it. And I'd just like to end with a big thank you to all of the Saunders Lab group members, especially Cesare and Diane, for being so welcoming and helping me out throughout. And it was such a valuable opportunity. And also a big thank you to the RSB. It's been so great to reflect on how all my work has developed. And I've put my contact details at the bottom. So if anyone has any questions, do feel free to reach out. But yeah, thank you so much.